bien, bienvenue à tout le monde pour cette séance organisée par la 6e section vétérinaire. Et je passe tout de suite la parole à Vanessa.
Merci, Monsieur le Président, professeur, docteur, mesdames, messieurs, et puis tous ces nouveaux visages que l'on voit aujourd'hui avec nous, les nouveaux membres de l'Académie des Jeunes. Eh bien, bienvenue à tous. Alors, comme toujours, pour ceux qui y sont habitués, 10 minutes seront consacrées aux échanges avec l'orateur après chaque conférence. Les académiciens qui le souhaitent, et donc vous aussi, vous pourrez poser vos questions. Les académiciens qui sont sur Zoom peuvent le faire en live. Et je vois qu'aujourd'hui, nous avons des gens qui habituellement sont sur Zoom et qui sont aujourd'hui ici. Donc, nous aurons peut-être moins de questions sur Zoom. Vous allez voir donc le message habituel. Ah, il est là. Et vous allez voir le message habituel s'afficher sur votre écran et ça nous permettra de vous donner la parole. Si vous nous suivez sur YouTube, merci d'écrire votre nom, votre prénom et votre institution et bien entendu votre question. So, the first lecture today will take us into the fascinating world of the Atlantic herring, one of the most abundant vertebrates. Thanks to its high fecundity, its diversity of ecotypes, and its early exposure to environmental conditions, this species provides a unique opportunity to explore the genetics of ecological adaptation in vertebrates. Mr. President, you will now introduce Professor Andersson, who will guide us through this topic.
Thank you. Professor Leif Andersson is a senior professor in functional genomics at Uppsala University. He is also professor of animal genomics at the Texas University College Station. He is member of the Royal Swedish Academy of Sciences, member of the European Molecular Biology Organization, and foreign associate at the National Academy of Sciences in the United States. He is also Doctor honoris causa of the University of Liège in Belgium. He is awarded with several prizes, Linné Prize in Zoology, Wolf Prize in Agricultural Sciences, Björkenska Prize in Medicine at Uppsala University, and the Grand Prize 2025 of the Finnish Society of Sciences and Letters. We are listening to you.
Thank you very much. It's a real honor to be invited to the Royal Academy of Medicine and talk about herring. And I'm really pleased to see that so many have come just to hear about the herring and what we can learn from that. So let's start with this picture, which is in fact from a children's book published in Sweden, which tells about the importance of the herring in the ecosystem. So the really important thing here is that herring is highly abundant, and it is the link between primary plankton production and higher trophic levels, which could be the cod, for instance, or human fishing. So I also would use some French in my talk at least. So this is a citation from the 18th century. And what I think, so ecological importance is very clear about the herring, but also you can't overestimate its economic importance in Northern Europe, that herring fishery all the time since the settlement of the Northern Europe had been really important for food security and economy. And so the work we're doing has also a practical implication. So I would argue that herring is an ideal vertebrate model to explore the signatures of selection. And one reason it's a fish. And what I mean by that, I think it's much more ecological adaptation in a fish than in a bird or a mammal. And I hope that my talk will give a clue why that is the case. So this is very important. So herring is a demersal spawner, which means that it has sticky eggs that stay on the spawning ground, either on gravel or seaweed. So that means that the whole development takes place at the place where they are spawning. And then they have a strong homing behavior, similar to salmonids. They go back to the place where they were born. And that means that leads to subpopulations spawning in different areas. So you could say that the population structure of herring is determined by when and where they are spawning. So what we have then is subpopulation that we often call ecotypes that have ecological adaptation. Then it's a broadcast spawner, which means that when they are spawning, there are huge numbers of females and males that is releasing sperm and egg. So it's perfectly random mating in the population. It has very high fecundity, so a female can produce 20,000, 30,000 eggs during a spawning. And that means that there's a lot of room for natural selection to select the most fit individuals. And then it has this huge population size. You know, when we study the genetics of populations, and humans is a good example. So if you compare different ethnic groups of humans or from different geographic areas, we see all these changes in allele frequency, which is just due to what we call genetic drift. So chance events from generation to generation. And the herring is very different. It's this huge population and also some gene flow. So when we look at what we call neutral polymorphisms, those which doesn't affect the phenotype of the organism, we see no difference whatsoever across the Atlantic Ocean. Another important factor is that herring has recently, within the last 8,000 years, adapted to a new environment, and that is the brackish Baltic Sea. So before that, there was glaciation on top of that. The glacier melted away, and it opened up the Baltic Sea. And from 8,000 years, it has been a brackish environment. So I also work on Darwin's finches on the Galapagos, and I used to say that the Baltic Sea is our Galapagos, because within the last 8,000 years, a number of organisms have adapted to live under these brackish conditions, low salinity. And it's good to eat. So another way to explain this is to say that if you really would like to study natural selection, so an experiment you can do, you find an infinite population, and then you split it into multiple infinite subpopulations. And then you expose this to different environmental conditions. Because what will happen then is that only the genes under selection will change in frequency. Everything else is constant. And you could say that herring is as close as you can come to that situation in a vertebrate. So what can we study in the herring? We can study adaptation to variation in salinity, obviously, but also climate, because it's distributed from basically this part of the Atlantic all the way up to Greenland and in the Arctic. And we also have photoperiod regulation of reproduction, which means that you have that herring spawns from winter to spring to summer to autumn. So, and we have shown that this is to a large extent genetically determined. The light conditions differ a lot between Baltic and Atlantic, feeding behavior, metabolic regulation. So there's a lot of interesting biology that you could study using this model. So what we have done, so I should say that, in fact, herring was my thesis project when I was a student more than 40 years ago. So I sampled herring and did some genetic analysis in the late 70s and found no difference whatsoever because of this, that neutral polymorphism doesn't show. So when we have screened for genetic diversity, some of the samples, so for instance, this sample up in Kalix, which here the salinity is 2 to 3 per mil, almost fresh water, because the river flow into that bay. So that's a sample there, I think 1979, and we have been using this in our work. But recently we have sampled many areas. So you see the Baltic Sea, this transition zone, and Kattegat, Skagerrak, the North Sea. And no sample from Belgium yet, but we have from English Channel here. So, and Iceland, Greenland, and also in North America. So, and then what we have done is we have generated a genome assembly, and then we do whole genome sequencing. All these populations have been done by whole genome sequencing. And then we study the genetic relationship. So what we could say, this is the resolution that we get when we compare their genetics. So these are the herring from the Baltic Sea, and they are split into the autumn spawners and spring spawners. They are clearly, so this is a principal component analysis that summarizes the genetic relationship. And Atlantic Ocean have the same thing, spring and autumn. And then you have the transition zone, Norwegian fjord, and this is the area around Ireland, Britain. I would guess if you sample herring at the Belgian coast, I would expect that it will fall into this group here. But what is important to notice is that even within this group, there are genetic differentiation. So this is just a broad picture. And what I would like to mention is that this has also led to genetic tools for efficient stock assessment. So so herring is one of the 10 largest marine fisheries in the world. So it's still a very, very important fishery. And there's a lot of fishing going on in the North Sea and the Norwegian Sea here. And populations, so I bet that the herring spawning in Belgium will migrate out to the North Sea for feeding. So in that, different populations are mixing. And the tools that we have developed now is the best method to separate them and monitor the development of the different stocks to avoid that you overfished, and which is a major problem. So our sampling strategy is illustrated here. So here you have this contrast between the fully marine environment in the Atlantic and the brackish environment. And I mentioned that the salinity up here is 2 to 3. It's 6 per mil in Stockholm, and then down here it's about 10 per mil. And then it's a major shift when you go through Öresund out here, and here you have 34 to 35. So what we do is that we take all the samples from the Baltic Sea, and then we have population samples from the Atlantic Ocean. And then we ask the question, what are the difference between the Atlantic population as a group versus the Baltic population as a group? So these are the differences. And then what you find on this axis, this is the chromosomes of herring, and this is the significance. And in this study, we used like 6 million sequence variants in this analysis. And almost all are at the bottom here, 0. But each dot here is one single sequence variant. And you see some show very striking differences. This is rhodopsin, for instance, which has to do with light vision in the Baltic Sea. So, but then you could, what we really would like to do is to understand how do these, so rhodopsin, we know that contributes to vision in the Baltic Sea. But we really would like to understand what these other loci are doing. And it's challenging because it could have, which life stage does a certain gene is important for, and so on. So then what we have done recently, we have used Ringkøbing Fjord as we call it our Rosetta Stone. And the reason for that, so Ringkøbing Fjord is an interesting case. So this is the west coast of Denmark. So these are Atlantic herring that spend all their life in fully marine environment. They only come in, so Ringkøbing Fjord itself is brackish. So it come in there for reproduction. And then when the larvae is a bit older, they leave and go out and feed on the plankton production in the North Sea. So what that tells us is that, and what we have seen is that some of these genes that is selected in Ringkøbing Fjord is also selected in the Baltic. And that tells us that those variants must be important for reproduction in brackish environment. So this comes back to this story now with the homing behavior. So remember now, there's a lot of different Atlantic herring. Some reproduce in fully marine environment. But if they reproduce in this brackish fjord, they will come back there year after year and has adaptation to survive there. So this is this contrast that we did. So on the upper side, these are the loci that we see when we compare Atlantic versus Baltic. But the mirror image is Atlantic versus Ringkøbing Fjord. All the red dots here and all the red genes are genes that replicate in Ringkøbing Fjord and in the Baltic Sea. So that tells us that these should be important for reproduction or early development. And that has led to this discovery that we just finalizing the paper describing of 4 critical loci. I don't have time to talk about all, but I will talk about one of those. And one is this gene here, which codes for an ion channel that is sperm specific. So this is called volume-regulated anion channel, and it's likely that we have those anion channels in all our cells, and they're really important to avoid that the cells are swelling. And this is a specific isoform that evolved in the clupeids and is contributing to avoiding swelling of the sperm in the herring. Here we have 2 genes that affect the envelope. So one is a structure protein, which is a sonapeloside protein, and an enzyme that make crosslinks and make the eggshell harder. And this is this little man here. They are working they use this silver tape to make it stronger, to make the surface stronger. And the effect of this is very, very important, because so you get a more dense eggshell in the Baltic Sea. And so this is experiment we did this spring together with Arild Folkvord at Bergen, is that we incubated eggs from Atlantic herring here and from Baltic herring here. This is marine environment, this is like a transition zone, and this is the Baltic Sea. And you see that the Atlantic herring eggs are swelling in the brackish water, which would be really bad for the development of the embryo, whereas the Baltic herring is protected from that swelling, and that is due to the effects of these two genes, a structured protein and a transglutaminase. So this transglutaminase is in fact related to coagulation factor 13A, that is really important for making crosslinks during coagulation. Then we have this problem that you have this little fellow that would like to hatch, then he has a denser shell to go through, so he need more molecular scissors to get out of this. And he has the high correlative, or she, they have the high correlative enzyme. And this is what they call fish hatching enzyme. So it's a metalloprotease belonging to the astaxanthin superfamily. And this is expressed exclusively in the hatching gland cells of the embryo. And what is fascinating is that this enzyme breaks down exactly those crosslinks that the transglutaminase is making in the Zona pellucida protein. So you have three factors that together cause a stronger egg envelope and then an enzyme that breaks up the egg envelope. So this illustrates the very strong selection pressure. So we call this a heat map, and the columns here, so one column is one SNP. These two are missense mutations. These two first rows are Pacific herring. These are Atlantic herring here, and these are herring in the Baltic Sea. And what you could see is that the Baltic herring has a Pacific haplotype. And Ringkøbing Fjord has the same Pacific haplotype as the Baltic herring. So this sounds crazy, right? That how could genes from Pacific herring find their way into the Baltic Sea, so far away from San Francisco to Stockholm? The thing is that the Pacific herring has colonized the Arctic Sea. So if you go to White Sea and the Pecora Sea, what you find is that there is Pacific herring. So Pacific herring and Atlantic herring meets about the border between Norway and Russia. And we have already demonstrated there is some gene flow across that hybrid zone. And apparently, it makes sense that there is this integration, because the Pacific herring has a different spawning behavior compared with Atlantic herring. Atlantic herring is spawning at deeper water, whereas the Pacific herring goes into estuaries, more shallow water, where it's often low salinity. So they were adapted to that type of spawning behavior, and therefore this variant was successful in the Baltic. So what we have done recently is to explore this in some more detail, and so we have used PacBio sequencing to sequence this region there. And what we find is this dramatic amplification of copies of this fish hatching enzyme. So these are haplotypes from Baltic herring, these are from Atlantic herring. So normally you expect to find about the same genes if you compare two alleles from the same species. But what you find here is that the Baltic herring, so blue here is the fish hatching enzyme, that they have about 20 extra copies of this. And when we study this across population, we see this remarkable correlation between copy number and salinity. So the Atlantic herring has basically no copies here, and whereas the Baltic herring has a lot of copies, and you see intermediate levels in the intermediate zone. And together with colleagues in Japan, we have measured enzyme activity from the matings we did in the spring. And if you measure enzyme activity of this enzyme, this is brackish water salinity, and you see there is 7-fold higher enzyme activity in the Baltic allele than in the Atlantic. But there is less difference at higher salinity, which is like the... and this is relative activity, and you could see that the fish hatching enzyme from Baltic herring has a peak at low salinity, whereas the Atlantic has a peak at higher salinity, which means that it's not just a copy number, but it's also the sequence of those which has an impact and affect the peak of activity in relation to salinity. So I think this is a beautiful story, but we also have other things that we could look at. So adaptation to climate. So in this case, we do a similar contrast, but now we don't contrast North versus Baltic versus Atlantic. Now we compare North versus South. So these are the populations that are spawning in Greenland, Iceland, part of Norway, compared with those in English Channel, Irish Sea. And what we find then is that we find much fewer regions of the genome. These red dots here are, in fact, inversions, big flops of chromosomes that contribute to adaptation to climate. And we think that these are the ones that will be important for future climate change, for the species to adapt to different environments. So something which I think is fascinating here is that when we do this type of analysis, many of the genes that we identified has in fact really important implications for human medicine. So if you look at the signals we have here, we have one is THR beta. This is estrogen receptor. This is a solute carrier protein, which is also important in human. And this is one which is particularly important. That is aryl hydrocarbon receptor, which is thousands of papers on toxicology. So this is the reason why humans, mammals, other vertebrates are so sensitive to dioxin and other pollutants, is because they bind this receptor that which goes into the nucleus and change gene expression. So that's really important. Okay, now I have another thing that's really important to study is what's called photoperiod regulation of reproduction. So all vertebrates has a mechanism how to sense changes in day length and adjust reproduction in relation to day length. So if you, for instance, if the optimal conditions for the offspring occurs in the spring and your gestation time is 6 months, then you should mate 6 months before the optimal. So this mechanism to sense day length through melatonin secretion, and this is only partially understood. So it's a part of the brain called pars tuberalis. What is key in this pathway, downstream of melatonin secretion, is the secretion, expression of TSH, thyroid stimulating hormone. And that seems to be key both in mammals, birds, and fish, the upregulation of TSH, and it activate here. And what we have done in the herring is that we compare spring and autumn spawning herring. And interestingly, the strongest signal that we always see is a gene called thyroid stimulating hormone receptor. And so to explain this, so here we do the contrast between spring spawners and autumn spawners in the Baltic Sea. Here we replicate the same question, but in the Atlantic Ocean. And I think it's just this remarkable replication that you see the same loci pops up in the two contrasts. If you now zoom in, so this is chromosome 15 in herring. If you now zoom in on chromosome 15, you see it's not one signal, but it's multiple signals. So what you have here, you have SOX11, TSHR, calmodulin, and estrogen receptor. And if you compare that, so this is in the Baltic, and if you compare that in the Atlantic, you see exactly the same pattern here with multiple peaks. And we think these are the genes which are in the selection. And this is what we really would like to understand. So let us zoom in a little bit more. So let us zoom in on SOX11B. So this is, in fact, a slide that made by Li Yingtang, which is a new postdoc in my lab. He did a PhD early this year in Liège with Carole Chalier as a supervisor. And so this is, and his project with me will be to really try to understand the molecular mechanism regulating reproduction. So here you have SOX11B. We have no, so you see there is no signal at the gene itself, but about 50 kb upstream, we have this very, very strong signal with two SNPs that stands out far. You see the p-value is 10 to the minus 200 almost. So these two SNPs is good candidate to be causal SNP, to have a role in regulating SOX11 in this pathway. So what we're doing at the moment is to use our PacBio sequencing to see, because this is just based on single nucleotide changes. Now we would like to see if there are any insertion, deletions, copy number variation, as we saw for the fish hatching enzyme. And I would like to end by saying about a new project that we have started, where the ambition is to, we have these wonderful signals of selection. So we have done these genome scans and we have identified regions of the genome that have contributed to different, but the task we would like now is to go into the molecular mechanism. And the key to that is a new fish laboratory that has been established in south of Stockholm, where we can now do matings with herring and keep herring and measure gene expression. So our ambition is to use the most advanced tools used in human medicine to explore the molecular mechanism, and both with regard to salinity, climate adaptation, and regulation of reproduction. So that is something that I really look forward for the coming years. So thank you very much.
Is there any question in the audience? Yes, please.
Thank you for the talk. Did you also check if there is adaptation correlated to water pollution?
Yeah, that's a very interesting question. So with a real hydrocarbon receptor, I'm pretty sure that there's nothing to do with pollution, because the variant, so there's in this locus, there's one SNP that stands out as the most strongly associated, and that is a missense mutation in this receptor, and the most common type in southern distribution is the same sequence as in human, in fact. So the variant allele occurs in the coldest water, so which is also the least polluted waters. So we think this is more an adaptation to protein-protein interaction involving this receptor. But it's possible that pollution could affect the frequency of these variants. And that is in fact something we are looking into, because the Baltic Sea has this major problem with pollution. And it's possible that it is affecting this genetic polymorphism. So that's something we are looking into at the moment. So.
Cédric Blancpain.
Fascinating talk. I've two questions for the price of one. The first, did you look at Catadromus or Anadromus fish to see if you see the adaptation like you observe in the different population of earring? If the gene duplication, change of enzymatic activity?
You know, so what it's important to, that's a good question. So it's important to realize that. So what's special with herring is that it managed to spawn and be a dominating species in an environment where salinity goes from 34, 35 to 2 to 3 per mil. So many species has this. So my view is that adult fish is very plastic. They can go between salinities. But the reproduction is the really challenge, to be able to reproduce in a difference. So salmon goes between, but they always reproduce in fresh water. Eel is the opposite. It spends most of its life in freshwater, brackish water, but always go to marine for reproduction. So I think, but certainly, you know, I think it will be really interesting to look at this in other species that has. So we have other species that adapted to the Baltic Sea, and I think it's quite clear that sperm biology and egg biology is really important, but it hasn't been studied in such a detail as we come to here. So it should be interesting to study. Yeah.
And the other question, if I may. You mentioned that in the very same place you have fish from the same species that spawned in the spring or in the autumn, right? So because that's a key question in evolution. If you want to make two different species from one, you should avoid them from crossbreeding constantly, otherwise you mix up the diversity. Yeah. So do you think that this will lead to two species on a speciation of two independent species?
I think you know, so what is interesting, so this spring autumn is replicating. So it's replicating in the Baltic. You see it in the East Atlantic. You see this in West Atlantic. So the variance, and we have also studied this TSHR region. So there is a quite large region that differs between the spring spawning and the autumn spawning. And we estimate that that haplotype has evolved at least over 500,000 years. So the two haplotypes, one is better adapted for, so my feeling is that there is an ongoing gene flow between these two, but then you have this strong selection acting on top of that. So they're really well separated genetically, but they are not completely reproductively isolated. And you know, we see that also gene flow between Pacific herring and Atlantic herring, they split 3 million years ago, so which is a bit unusual to have gene flow between species that have been separated for such a long time.
To cross them.
Yeah.
To cross them.
Oui, il y a une question là-bas dans le fond, et le micro devrait arriver à Cédric Blancpain dans le fond de l'auditoire, s'il vous plaît. Madame, allez-y. Yes.
Okay, yeah. Could you speak up, probably here?
So, beautiful talk. Two small questions. One was what caught my attention in the hits that you have were endoplasmic reticulum genes. I saw SEC16B and HERPA2. So do you have any idea what's being secreted and the potential role of ER protein synthesis, processing, and exit? Yeah. And the second question is related to SOX11B. So you have, you're looking at that because that's the nearest gene, but are you considering EQTL approaches or something to actually demonstrate that this is indeed the gene that's involved?
Yeah, in this short talk, you know, SEC16B is a really interesting signal that we have, and you know, so this is an example of a gene. So this is when we compare spring and autumn spawners, but this signal we only see in the Baltic Sea. We don't see it really in the Atlantic Ocean. So we don't think this has anything to control reproduction, but we think this is a variant that is important for being a spring spawner in the Baltic Sea. And so 616B is really interesting from a human medicine point of view, because I don't know if you're aware of that, but that if you look at this GWAS analysis in humans, so this is the most consistent signal for juvenile obesity in humans, so, and also age at menarche. And I think there are more than 150 papers that highlight SEC16B and related to metabolic and lipid metabolism and so on. So what is interesting with, if you compare the Baltic with Atlantic, is that Baltic is much less, so the fish in the inner Baltic is much smaller than in the Atlantic. And they're much more lean, they have less, and we think this is because of the plankton production is different in the Baltic Sea compared with the Atlantic Ocean. So there's something with handling lipids under these limiting levels. So I think I've argued, in fact, that this is a model to understand, because we don't really understand this signal in the human medicine yet. So we hope to, and this is in fact one of the loci that we will study in this new project. Then was the SOX11B. Yeah, absolutely. So what is so SOX11 is one of these transcription factors that is very pleiotropic, do many things in different cell types, and often is present in a gene desert. So you see there is only one gene here, and then, so at present, I mean, we can't exclude the possibility that in fact this is affecting these genes here, but I bet that this is the gene that is under selection here. And what's typical for this transcription factor is that they have long-range regulatory element, and I think that is what is going on. So this is really what we would like to do in this new project, where we plan to do single cell sequencing, attack sequencing, and spatial transcriptomics, are the different stages of reproduction, to figure out how is this doing. Does this crosstalk to the TSHR locus, for instance, and try to understand how this is regulated. So, yeah.
It was a fascinating talk. So I have a question concerning the loci you find to be selected with the change in salinity. So if one would, with a naive view of physiology, one would expect that the change in the cell volume would be, more regulated by ion channel, transporter, and water channel, rather than simply the strength of the membrane rupture that would occur following this change in the volume. So you haven't seen any kind of polymorphism associated with one of those regulator of water flow?
In fact, we have signals of selection also on solute carriers and aquaporin, in fact, in this one. So, but we have not yet finalized the full characterization of those, and the plan is to do that also. So in the spring, we plan to take in and redo this and expose them to different salinities and continue to do. So I'm sure that this thing, the constitution of the sperm and egg, is really important, but it's not the only aspect of it. So I'm sure there will be a continuation of this story. So, in fact, you saw I was a little bit surprised to see. Let me see. Okay, I'm going the wrong direction. So here you have one of those solute carriers. I was a bit surprised to see them here in the north-south contrast, because we see the same gene in the contrast Atlantic-Baltic. So.
No other question in the audience?
Almost everyone is here, so last question on the internet. Thank you. Thank you, Professor.
Thank you very much.
Thank you very much. «
25 à 50% de... Oh, pardon. Je suis trop loin, je remonte, c'est pas le bon chapeau. Selon l'Organisation mondiale de la santé, l'OMSA, plus de 75% des maladies infectieuses émergentes et réémergentes humaines sont d'origine zoonotique. L'Europe centrale est particulièrement exposée au risque de voir apparaître de nouveaux virus, les arbovirus favorisés par le réchauffement climatique, des virus à transmission non vectorielle ainsi que des virus émergents beaucoup plus inattendus. Monsieur le Président, le professeur Zientara, membre étranger de cette compagnie, va nous en dire plus dans un petit instant.
Oui, merci. Nous avons le plaisir donc d'accueillir Stephan Zientara, qui est docteur en médecine vétérinaire, et qui a obtenu l'habilitation en 2001 et qui est directeur du laboratoire de santé animale de l'ANSES à Maison-Alfort en France. Il est responsable du laboratoire de référence de l'Union européenne sur les maladies équines et responsable aussi d'un laboratoire de référence de l'Union européenne sur la fièvre afteuse. Il est expert en santé animale auprès d'organisations internationales comme l'Organisation mondiale de la santé animale, la FAO, l'Organisation pour l'alimentation et l'agriculture, l'Autorité européenne de sécurité des aliments et la Commission européenne sur les maladies virales. Et nous allons donc l'entendre sur les zoonoses virales émergentes en Europe centrale. Merci.
Thank you, Mr. President. Thank you very much for this invitation. As my French is much better than my English, as I'm French, as I'm here in Belgium, I will give my talk in French. Donc le sujet qui m'a été demandé de présenter s'intitule « Les zoonoses virales émergentes menaçant l'Europe centrale ». Et pour tout dire, c'est Claude Sagerman qui me l'a présenté. J'espère qu'il me remerciera, ou je sais qu'on verra après. Donc tout d'abord, l'Europe centrale, et en fait quand je me suis intéressé à cette définition, c'est une région finalement qui a un contour assez flou. Et donc globalement, les géographes et géopoliticiens s'accordent à dire que ça doit correspondre à peu près à la liste de ces pays, mais vous trouvez une définition des fois un peu différente. Pour ce qui concerne donc les zoonoses, comme ça a été dit, il y a beaucoup de maladies infectieuses émergentes humaines qui ont une origine animale, et vous avez quelques exemples ici. Le dernier étant le SARS-CoV-2, par exemple, assez connu. Et il y a une liste très longue de virus qui sont capables de franchir la barrière des espèces ou qui ont déjà franchi la barrière des espèces. Donc évidemment, je ne serai pas exhaustif parce que là, il ne faudrait pas 20 minutes, mais probablement en 3 jours. Et donc, je vais donc en sélectionner quelques-uns. Peut-être un élément important que nous allons vivre dans les années qui viennent ou dans les décennies qui viennent, c'est le réchauffement climatique. Et comme vous le savez, la température du globe a augmenté et va dépasser d'ailleurs le seuil fatidique des 1,5 degrés du traité de Paris. Là, je vous ai mis les données de Copernicus des 3 derniers mois de septembre 2025, 2024, 2023. Et vous voyez que ces températures sont très élevées par rapport à ce qu'on a pu observer depuis 1940, même si tous les hommes politiques ne sont pas complètement convaincus par ce type d'argument. Toujours est-il que j'ai pris quelques exemples strictement en virologie humaine, et après je reviendrai sur les zoonoses. Mais si vous prenez le cas, par exemple, du moustique tigre Aedes albopictus, il est arrivé en France en 2004, du côté de Menton. Là, vous avez la situation en 2015 et vous avez la situation en 2025. Moustique qui est en train de s'étendre et qui est capable donc de transmettre plus de 50 virus et qui a cette compétence. Vous avez la situation ici de ce virus en 2010 et vous avez de ce moustique et vous avez les projections ici en 2030 comme vous pouvez donc le voir ici, et donc une extension de ce moustique en Europe et notamment en Europe centrale. Le 13 juin 2025, on a eu le premier cas autochtone en France de chikungunya qui a été décrit dans le sud de la France. Mais depuis, au 13 octobre, on a plus de 770 cas de chikungunya et de dengue, autres virus transmis par des moustiques, qui sont essentiellement des virus de pays chauds. Et pour la première fois, dans des régions comme la Bourgogne-Franche-Comté, le Centre-Val de Loire, le Grand Est, et aussi la Nouvelle-Aquitaine, ce qui est complètement nouveau. Et vous avez ici l'aire de répartition de ces infections, en particulier Île-de-France et l'Est. Et là, je vous ai rajouté les infections à virus West Nile, dont je reparlerai, 48 cas humains, avec pour la première fois des cas en région Île-de-France et en Auvergne. Donc on voit vraiment qu'il est en train de se passer quelque chose. Et ce que l'on voit ici en France va très probablement évoluer dans les années qui viennent et atteindre le nord et l'est de l'Europe. Alors d'ailleurs, nous avons créé avec deux collègues, donc Isabelle Delambary, qui est professeure de virologie médicale à Marseille, Annabelle Afayou, qui est entomologiste à l'Institut Pasteur, et moi je suis le vétérinaire de service, Arbo France, qui est une structure qui a pour but c'est de stimuler la recherche sur les maladies à transmission vectorielle. Et nous avons d'ailleurs aussi une société européenne d'arbovirologie. Donc, pour ce qui concerne les prédictions, vous savez que la prévision est difficile, surtout lorsqu'elle concerne l'avenir, comme a dit Pierre Dac, mais c'est probablement d'ailleurs Mark Twain qui a dû le dire. Et donc je vais quand même essayer de me livrer à cet exercice en prenant peut-être deux types de zoonoses, les zoonoses qui sont déjà plus ou moins présentes en Europe centrale ou à la frontière de l'Europe centrale et qui vont très probablement s'étendre dans les années qui viennent, et puis des zoonoses qui ne sont pas encore présentes mais qui ont un vrai risque d'introduction dans le futur. Alors on va commencer par le virus West Nile, qui est un orthoflavivirus transmis par des moustiques. Il y a 9 lignages de ce virus qui ont été décrits. Lignage 1 et 2 infectent l'homme et les chevaux. Il y a récemment eu une publication américaine qui montrait qu'un virus de lignage 3 était capable aussi de neuroinvasion chez l'homme. Donc on surveille ce lignage 3. Mais est-ce que c'est un phénomène anecdotique ou est-ce que ce sera plus important ? Toujours est-il que ce sont des virus orthoflavivirus transmis par des moustiques, notamment des moustiques du genre Culex, des femelles. Et ce virus circule parmi les populations d'oiseaux, notamment les oiseaux migrateurs, moustiques, oiseaux, et de temps en temps, ces moustiques vont aller piquer des autres qui sont des cul-de-sac épidémiologiques, l'homme, le cheval et de nombreuses autres espèces animales, sachant que l'homme et les chevaux ont le même tableau clinique, avec parfois des neuroinvasions et des encéphalites et méningoencéphalites qui peuvent s'avérer mortels. Alors, ce virus, on en a parlé, il a été isolé en 1937 en Afrique, sur la côte ouest du Nil. Et puis surtout, il est arrivé donc, aux États-Unis, à New York, en 1999. Et vous avez aux États-Unis à peu près entre 120 et 125 décès par an, avec un nombre important donc, de gens qui ont été infectés. Et vous voyez plus de 30 000 formes neuro-invasives, donc des gens qui ont des atteintes neurologiques sévères, uniquement aux États-Unis. Il est maintenant présent sur tout le continent américain. Et on a évidemment aussi des cas chez les chevaux. On a exactement la même évolution. Une des différences étant qu'il y a un vaccin chez les chevaux, ce qui n'est pas le cas chez l'homme. Toujours est-il que si on revient sur l'Europe, évidemment, les mouvements liés aux trajets migratoires jouent un rôle important. Et avec le réchauffement climatique, ces trajets sont modifiés, les oiseaux restent plus longtemps, il y a des cycles réplicatifs plus importants, et ils vont dans des régions, notamment au nord de l'Europe et dans l'Est. Et s'ils sont affectés, ils amènent avec eux donc, ce virus. Donc en Europe, on a eu à partir des années 98 l'arrivée du lignage 1, 2004 l'arrivée du lignage 2, donc on a 2 lignages qui co-circulent, 1 et 2. Et je vais évidemment pas le temps de tout décrire, mais on a eu les premiers cas humains en 2019 en Allemagne et aux Pays-Bas en 2020. Vous pouvez voir ici sur la carte. Et je me rapproche très rapidement en 2022, en France, où nous, on avait du virus toujours dans l'axe méditerranéen, entre l'Espagne et l'Italie, depuis les années 2000, très régulièrement. Et on a vu des cas arriver cette fois-ci du côté de Bordeaux, chez les chevaux, qui ont été très utiles pour permettre de mieux surveiller la circulation de ce virus et dire d'ailleurs aux autorités en charge de la transfusion sanguine de bien surveiller les donneurs dans ces régions. Donc ce sont des données vétérinaires qui ont des retombées sur la surveillance, notamment dans le domaine des greffes ou de la transfusion. 2023, il y a eu des cas humains, des cas chez les chevaux, des cas chez les flamants roses d'ailleurs du zoo de la Palmyre. Donc là, on a une installation vraiment du virus West Nile. Donc pour vous montrer encore une fois que ce qu'on a vu en France va probablement aussi se décliner dans le futur, dans la partie centrale de l'Europe. 2024, voilà la situation qui était celle donc de l'Europe, hein, avec de nombreux cas humains, chevaux, et on voit déjà en Allemagne, notamment en Autriche, une concentration de cas très importante. Et 2025, on a eu la première mise en évidence de ce virus en Seine-Saint-Denis, donc au nord de Paris. C'était la première fois qu'on voyait ce virus avec des cas autochtones, donc des gens qui n'avaient pas quitté la région parisienne, pas des cas importés. Donc voilà la situation. Donc probablement que ma carte est fausse, elle date d'hier, mais ça change tous les jours. Donc là, on est à 48 cas humains. Il y a 2 cas qui vont probablement être confirmés bientôt. 56 cas équins, et vous avez les cas aviaires en fonction donc, des couleurs. Et vous voyez, le sud de la France est normal. La Nouvelle-Aquitaine, c'est quand même plus surprenant. Enfin, depuis 2 ans, on a ça. Le centre de la France et la région Île-de-France. Et donc voilà un peu la synthèse. Et je vous ai mis en rouge les départements pour lesquels on a pour la première fois identifié ce virus, et notamment donc, à Paris, donc des chevaux de la garde républicaine dans Paris et autour de Paris, Orléans par exemple, donc ce qui est quand même assez étonnant. Et là, vous avez les cas humains, les cas autochtones dans la région Île-de-France, soit identifiés à partir de manifestations cliniques ou dans le cadre du dépistage pour des dons de sang. Donc voilà la situation en Europe et des éléments qui montrent qu'il y a bien une évolution, mais vous le savez mieux que moi puisqu'en Belgique, il y a eu les 3 premiers cas identifiés chez des oiseaux très récemment. Si on se rapporte à la situation italienne, il y a plus de 718 cas dans le nord de l'Italie, dont 49 décès. Et encore une fois, ces chiffres sont faux, puisqu'il y a encore, on est en plein dans le pic épidémique ou épizootique. Hein. La saison va s'arrêter probablement début décembre. On a eu récemment aussi, les collègues hollandais ont identifié ce virus chez un cheval récemment. Et donc, on a à peu près pour l'instant plus de 900 cas chez l'homme, avec une grosse partie d'ailleurs de cas en Italie. Donc là, j'ai un petit film, j'espère qu'il va marcher. Voilà, c'est bon. Et là, vous avez de 2014 jusqu'à 2025 l'évolution des cas de West Nile en Europe. Donc, vous voyez que c'était quand même assez localisé dans le sud de la France, sud de l'Europe. Et on voit maintenant une augmentation du nombre de cas dans le nord de l'Europe et en Europe centrale aussi. Ce qui est très différent de ce qu'on observait aux États-Unis, où vous avez vraiment une dissémination très importante, alors que nous, depuis les années 2000, on n'avait que des petits foyers épidémiques ou épizootiques, mais finalement, ces foyers sont de plus en plus nombreux. Et donc, si on fait une synthèse, 2014, on était dans cette situation-là, 2025, on est dans cette situation-là. Alors j'ai entouré en rouge l'Europe centrale, puisque c'était un peu la question qui m'était posée. Je ne dis pas qu'il y a un focus spécial sur l'Europe centrale, mais on voit bien qu'en Europe centrale, on a comme une modification aussi de la situation épidémiologique que l'on observait auparavant. Donc c'est un peu une synthèse donc, de ce que j'ai dit ici, avec le fait que ce virus se dirige vers le nord, vers l'ouest et vers aussi le sud de l'Europe. Alors cette publication est intéressante parce que finalement il y a relativement peu de travaux scientifiques qui montrent un lien complètement évident, démontré, entre réchauffement climatique et évolution des maladies, notamment des maladies vectorielles. Et cette publication est intéressante parce que les auteurs ont étudié tous les facteurs, la température, l'hygrométrie, le couvert végétal, la démographie, enfin tout un tas de facteurs. Et lorsqu'ils font uniquement bouger la température, c'est la ligne du haut, on arrive à des situations qui sont très similaires à ce qu'on observe vraiment. Et donc c'est une des publications qui montre le lien entre augmentation de la température et évolution épidémiologique différente des maladies vectorielles. Donc ça, c'était pour le virus West Nile. Un autre virus très proche du virus West Nile, c'est le virus Uzutu, qui est un petit cousin. Donc il a un cycle très similaire, oiseau-moustique, oiseau-moustique, et ces moustiques vont contaminer un certain nombre d'autres qui sont aussi des cul-de-sac épidémiologiques. Alors une des différences majeures entre le virus West Nile et le virus Uzutu, c'est que West Nile induit des encéphalites chez l'homme et chez les chevaux, globalement peu de mortalité chez les oiseaux en Europe, alors que le virus Uzutu, c'est l'inverse, très peu de mortalité chez l'homme, alors qu'il y a une grande mortalité massive chez les oiseaux, notamment les merles, les chouettes, etc. Donc là, vous avez une diapositive qui montre d'une part que ce virus a été isolé dans les années 59-60 en Afrique du Sud. Il tire son nom de la rivière Uzutu, au Swaziland. Puis il s'est répandu donc, dans toute l'Afrique et le reste du monde. Et vous avez un certain nombre de génotypes qui sont aussi présents en Europe. Alors, est-ce que c'est un risque en santé publique? Donc là, vous avez des identifications de génotypes qui ont été réalisées en Europe ces dernières années. Et surtout, ce virus a été aussi isolé chez l'homme, avec à peu près une centaine de cas chez l'homme, soit dans le cas d'atteintes neurologiques, soit dans le cas de découverte, de dépistage, dans le cas de la transfusion sanguine. Donc, il fait vraiment partie des virus que l'on surveille. On ne sait pas si c'est un phénomène, encore une fois, anecdotique ou c'est véritablement, dans le futur, il peut avoir donc, des conséquences majeures en santé publique humaine et vétérinaire, mais notamment humaine. Donc voilà un peu la localisation et les données sur la présence de ce virus, notamment, vous voyez, en Europe centrale, en Allemagne et les pays voisins. Alors, un autre virus aussi de la famille des flaviviridés, un flavivirus qui est le virus de l'encéphalite à tique, et dont, comme son nom l'indique, ce virus est transmis par des tiques, et l'homme peut être infecté lors d'une piqûre de tique, mais peut aussi être contaminé lors de la consommation de produits, notamment de produits laitiers non pasteurisés, où le virus peut être présent dans les fromages, etc. Donc, ce sont les deux modes de contamination de ce virus de l'encéphalite à tique. Alors, il a été montré que l'aire d'extension de présence des tiques en Europe augmente. Vous avez ici la situation des tiques pendant la période 2007-2009. Vous avez ici la présence de ce virus en 2022, et vous voyez que l'on voit une augmentation de la répartition de l'aire de distribution de ces tiques. Et je vous ai mis cet exemple-là. En 2020, on a identifié ce virus à partir de personnes qui ont présenté des méningites et des méningoencéphalites et qui avaient consommé du lait de chèvre non pasteurisé, donc transmission par voie alimentaire. Et là, vous avez cette carte qui vous montre la présence en 2003 du virus de l'encéphalite à tique, de l'incidence, avec une grande incidence, notamment, on en parlait hier, en Suède, dans le nord de l'Europe. Et on voit aussi qu'en Europe centrale, on a une augmentation de cette incidence. En 2022, il y avait plus de 3500 cas qui avaient été confirmés. Et vous voyez, on est à plus de 3000 cas officiels de cette infection en Europe centrale. Alors, un autre virus aussi, qui est cette fois-ci un virus à transmission non vectorielle, donc les virus influenza. Donc, comme vous savez, ces virus de la grippe influenza sont capables d'évolution génomique, soit par mutation de leurs différents segments, de leurs 8 segments génomiques, ou par échange de ces segments, ce qu'on appelle de réassortiment. Et ces virus peuvent aussi être transmis de l'animal à l'homme avec un cycle faisant le plus souvent intervenir les porcs qui ont les deux types de récepteurs. Souvent des virus d'origine aviaire peuvent être transmis aux porcs. Vous avez des types de récepteurs alpha 2,3, alpha 2,6 et contamination chez l'homme, même s'il peut y avoir donc, des transmissions directes. Et donc des schémas de transmission interespèces qui sont très complexes avec de nombreuses espèces animales qui peuvent être infectées, les cétacés, les félins, les visons, etc. Je ne vais pas en rentrer dans le détail, mais simplement vous avez ici une carte assez récente qui vous montre le fait qu'on a eu une évolution donc, de ces virus qui ont été identifiés chez de très nombreuses espèces animales de par le monde, sur le continent américain, mais aussi en Europe. Et cet article assez récent montre l'évolution, notamment du virus H5N1 depuis la période 2023-2019 jusqu'à une période un peu plus récente. Et vous avez ici des informations qui vous montrent que très régulièrement, on a donc des foyers de virus influenza aviaire en Europe, en Pologne, en Allemagne, etc. Donc ce sont vraiment des virus qui font surveiller. On voit l'augmentation de leur incidence et le risque potentiel de transmission à l'homme. Alors ça, il s'agissait donc de virus qui sont déjà présents, que l'on surveille, mais dont probablement les risques d'extension vont augmenter. Et puis j'ai pris quelques exemples de virus qui sont vraiment à risque d'introduction en Europe centrale. Ils ont déjà été introduits dans le sud de l'Europe, et donc il y a une probabilité difficile à calculer, mais qui est probablement non nulle, que ces virus puissent se retrouver un jour ou l'autre en Europe centrale. Alors un des petits derniers, c'est le virus de Crimée-Congo, qui est un bunyavirus, genre nérovirus très dangereux pour l'homme, avec 30 à 50% de mortalité, et qui est présent dans ces différentes régions du monde, Moyen-Orient, Extrême-Orient, Afrique, etc. Et assez récemment, des collègues, notamment les collègues du CIRAD et des gens de mon laboratoire, ont mis en évidence la présence de ce virus à partir donc de tiques collectés chez des bovins à l'abattoir, 2 abattoirs au nord et au sud de la Corse. Là, vous avez ici les segments génomiques, les séquences génétiques. C'est un virus avec 3 segments génomiques, LMS, et qui montre la présence de ce virus. Donc il y a des anticorps, il y a du génome. Ce virus a aussi été identifié en Espagne. Il y a eu des personnes décédées de ce virus en Espagne. Et donc on voit que ce virus a une aire d'extension qui est en train donc d'évoluer. Assez récemment aussi, cet article qui correspond à la mise en évidence de ce virus Crimée-Congo, mais cette fois-ci en France continentale, dans les Pyrénées-Orientales, des enquêtes sérologiques qui ont été effectuées chez des bovins, chez des chevaux, montrent que ce virus circule, ce qui n'avait donc jamais été mis en évidence jusqu'alors. Et puis autre dernière étude où les collègues ont analysé plus de 8000 sérums de bovins, plus de 2000 sérums d'animaux de la faune sauvage, des sangliers, des chevreuils, etc. Et donc, ils ont pu montrer qu'il y avait des animaux qui étaient séropositifs dans le sud de la France. Et donc, on a probablement la circulation à bas bruit de ce virus qui, encore une fois, a été complètement inattendu. Donc, il est possible que ce virus, dans les années ou les décennies qui viennent, puisse étendre son aire d'extension. Alors, il y a des virus qui sont aussi surveillés, dont les risques d'apparition sont beaucoup plus faibles, il faut bien le dire, mais ça fait partie donc des virus que l'on surveille aussi, qui sont des alphavirus, encéphalitiques indolèses de l'ouest vénézuélienne, qui sont présentes sur le continent américain. Donc ils ont des cycles assez complexes, faisant intervenir des oiseaux, mais aussi des rongeurs. Et ces virus sont capables d'infecter l'homme et provoquer donc des atteintes neurologiques chez l'homme. Donc là, vous avez l'aire de répartition de ces 3 virus, virus d'encéphalite de l'Est, de l'Ouest et vénézuélienne, notamment celui qui est là-bas à droite, qui est particulièrement zoonotique. Et donc ces virus, évidemment, entre le continent américain et l'Europe, il y a quand même un océan traversé, mais on a donc des systèmes pour surveiller notamment les chevaux qui sont importés d'Europe, des États-Unis, du continent américain, pour éviter d'introduire ce type de virus dans nos régions. Alors un autre virus aussi, qui est le virus de la fièvre de la vallée du Rift, qui est un virus présent sur le continent africain, avec des possibilités de transmission directe, par contact avec des animaux infectés, ou transmission par des moustiques en particulier. Et ce virus fait vraiment partie des virus que l'on surveille, car le risque d'introduction en Europe est loin d'être négligeable, et il pourrait éventuellement un jour ou l'autre se retrouver en Europe centrale, avec tous les éléments d'introduction que l'on voit. Alors si je prends l'exemple du domaine vétérinaire, on a des introductions aujourd'hui de dermatoses nodulaires contagieuses en France, enfin, en Sardaigne, dans le nord de l'Italie, en France, en Espagne. Là, on a des foyers à la frontière française, et la ministre de l'Agriculture vient d'indiquer le blocage de tous les mouvements d'animaux en France pendant au moins 15 jours. C'est quelque chose qu'on n'aurait jamais imaginé. Je ne parle pas de la fièvre catarrhale ovine, je ne parle pas de la maladie hémorragique épizootique, je ne parle pas de la peste porcine africaine. Donc, toutes ces infections dont on n'aurait jamais imaginé il y a encore une dizaine d'années, ou vingtaine d'années, qu'elles soient présentes en Europe, le sont. Et donc, ce type de virus fait partie typiquement des virus qu'il faut surveiller car le risque est réel qu'il soit introduit. Enfin, un des derniers, j'arrête, c'est le virus Europutsch. C'est un virus qui est cette fois-ci transmis par des culicoïdes, des moucherons, comme les virus de la fièvre catarrhalovine, MHE, etc. Donc en Europe, on a tout ce qu'il faut pour que ce virus puisse être présent en Europe. Et vous avez une photo de moucheron ici. Bon, il a un cycle assez complexe. Il est présent en Amérique du Sud, en Amérique centrale, mais par exemple en 2024, on a eu des cas importés en Espagne, en Italie 6 cas, Pays-Bas 1 cas, Autriche 1 cas, 3 cas en Allemagne et on a eu 6 cas en France. Et aujourd'hui, au niveau de l'ECDC et des autorités sanitaires françaises, italiennes, espagnoles, il y a des réflexions sur le risque d'introduction d'Europutsch. Et en ayant vu l'introduction de virus, par exemple comme West Nile, il faut vraiment s'intéresser à ce type de virus pour lesquels d'ailleurs on connaît très peu de choses, à la fois sur la biologie des vecteurs et les interactions entre ce vecteur et ce virus. Voilà, alors c'était quelques informations sur les risques potentiels que l'on peut connaître dans les années futures. Et donc je vous remercie pour votre attention.
Bien, la conférence est ouverte à discussion. Une question là? Oui.
Voilà, merci. Bonjour et merci Stephan pour cet exposé qui est évidemment très complet. J'aurais bien voulu revenir sur un point, c'est sur la relation entre l'élévation de la température et la distribution des vecteurs. Alors pour Aedes albopictus, le moustique tigre, qui est quand même d'origine plus tropicale en disant, on comprend qu'il y ait une relation, même implicitement une relation entre la progression du vecteur et la distribution plus large, en tout cas plus vers le nord, des maladies qui y sont associées. En ce qui concerne West Nile, qui est en fait dont le vecteur est un culex, le culex ce sont des culicidés qui sont présents dans toute l'Europe. Et moi je me posais la question, pourquoi est-ce que cette progression du virus West Nile vers le nord n'est-elle pas plus rapide, dû au fait que ce virus est transmis notamment par des oiseaux et notamment des oiseaux migrateurs, et plus rapide dans le nord de l'Europe que ce que l'on observe? Et est-ce qu'il y a vraiment un lien avec le changement climatique et la hausse de la température pour ce type de virus dont les vecteurs sont déjà présents de manière abondante dans l'ensemble de l'Europe?
Alors oui, tu as raison. Alors, déjà, l'équipe d'Annabella Fayou à l'Institut Pasteur a montré qu'Aedes albopictus, ce moustique tigre, était capable de transmettre West Nile et était compétent. Donc a priori, aujourd'hui, ce sont essentiellement les culex qui sont les vecteurs majeurs, mais il ne faut pas exclure quand même que le moustique tigre puisse jouer un rôle. Mais je suis d'accord avec toi, ce qui probablement joue un rôle, c'est plus les modifications des trajets migratoires des oiseaux et aussi les périodes pendant lesquelles ils restent présents. Ils restent présents beaucoup plus longtemps en France, en Europe. Il y a un beaucoup plus grand nombre finalement d'oiseaux qui vont migrer. Donc le risque d'avoir des oiseaux qui arrivent en Europe, qui soient virémiques, qui soient ensuite piqués par des moustiques, même s'ils sont présents depuis très longtemps. Les cycles réplicatifs sont une des explications, parce qu'on n'a pas d'explication claire pour être sincère, il s'agit par rapport à ce qu'on observe sur le continent américain. Pourquoi est-ce qu'on a eu cette explosion aux États-Unis et pourquoi est-ce que nous, on a des foyers, alors effectivement de plus en plus nombreux? Aujourd'hui, ce sont des hypothèses, mais on n'a pas de certitude.
Benoît Van den Einde.
Merci. Pour le West Nile, vous avez mentionné qu'il existait un vaccin pour les chevaux, mais pas pour l'homme. Est-ce que ce vaccin est efficace et est-ce qu'on peut s'attendre à un développement d'un vaccin humain dans un proche avenir?
En fait, il y a 3 vaccins chez les chevaux, 2 vaccins inactivés, un vaccin recombinant, qui sont commercialisés en Europe. Et chez l'homme, il y a donc des candidats vaccins, mais pour des raisons de marché, ce vaccin n'est pas n'est pas commercialisé. Donc après, je ne peux pas vous répondre sur l'avenir de ce type de vaccin, mais il n'y a aucune raison. En tout cas, chez les chevaux, ils marchent très bien. Et les données qui sont disponibles chez l'homme montrent qu'a priori, il a l'air de bien marcher. Après, ce sont des considérations plus financières que scientifiques.
Encore une question là au centre, dans le fond de la salle, et une autre à gauche.
Guillaume Dachy, hématologue, en tant que transplanteur. On est en train de doucement élargir les bilans sérologiques qu'on fait auprès des donneurs. Je m'occupe des transplantations de moelle osseuse. Est-ce qu'il y a des directives européennes qui vont être adaptées dans le futur en termes d'homogénéisation de cette check-up des donneurs en termes de possibilité d'infection des greffons?
Alors là, je ne peux absolument pas vous répondre parce que là, c'est vraiment hors de mon champ de compétences. Puisque moi, je suis vraiment impliqué dans la surveillance des maladies animales. Ce que l'on fait en France, en tout cas, c'est qu'on a des réunions régulières entre mon labo, par exemple, l'établissement français du sang, l'établissement français des greffes, et en fonction des résultats dont on dispose sur la surveillance, sur la circulation du virus dans les populations animales, on donne des recommandations aux collègues pour faire une surveillance sérologique, par exemple, ou génomique plus importante. Après, en termes de... Alors, il y a des réflexions au niveau de l'ECDC sur justement ce type de surveillance. Quelles vont être les conclusions? Je ne sais pas. Je sais qu'il y a des groupes de travail sur le sujet. Voilà, c'est tout ce que je peux vous dire.
Merci beaucoup pour ce brillant exposé. J'avais juste une petite question. En fait, je souhaiterais que vous puissiez proposer quelques recommandations en matière pour minimiser les risques et si c'est présent et que ça diffuse, quelles seraient vos principales recommandations?
De façon générale, essayez le plus possible d'avoir un maximum d'informations auprès donc, des pays voisins de l'Union européenne. Là, je vais utiliser plus un volet vétérinaire, mais là, on a eu un virus de sérotype 5 en Sardaigne qui a été identifié. On ne sait pas d'où il vient. De façon générale, tous les virus qui ont émergé en Europe ces dernières années, on ne connaît pas leur origine. Et ça, c'est quand même un énorme problème. Donc, renforcer ces collaborations, ce qui veut dire mettre des moyens financiers pour avoir des liens, des projets avec ces pays, ce qui est de moins en moins le cas, il faut quand même bien le reconnaître. Se préparer en ayant des outils de détection des vaccins, etc., puisque on l'a vu, alors pas dans le cas de West Nile, mais dans le cas d'autres maladies strictement vétérinaires où finalement il fallait vacciner en urgence, sauf qu'on avait très peu de données sur l'innocuité, l'efficacité des vaccins. Donc la guerre, si j'ose dire, se prépare en temps de paix, ou en tout cas en temps de paix, on essaie d'éviter d'avoir la guerre. Renforcer aussi les collaborations entre les différents secteurs, vétérinaires, humains, les gens travaillant aussi dans le domaine de l'environnement. Alors, c'est pas que du One Health un peu galvaudé. Dans le cas de West Nile, on a un vrai réseau One Health qu'on a d'ailleurs initié bien avant que le concept One Health existe. On a fait ça dans les années 90, où on mettait les gens qui étaient compétents en entomologie, en virologie humaine, en virologie vétérinaire, dans l'environnement pour étudier tout ce qui concerne l'évolution des populations de moustiques. Donc ça, c'est vraiment des choses qu'il faut mettre en place concrètement.
Oui, bonjour. Vous avez aussi en France des projets de recherche sur la lutte contre les vecteurs et notamment le relâcher de moustiques stériles, le relâcher de moustiques infectés par certaines bactéries. Qu'est-ce que vous pensez de cette voie-là en termes de prévention? Vous avez parlé des vaccins, mais la lutte contre le vecteur, est-ce que vous pensez que c'est aussi une voie importante?
Oui, alors oui, d'ailleurs c'est utilisé dans certaines îles françaises dans l'océan Indien, notamment le lâcher de moustiques stériles. Ça a l'air de marcher pas mal, même si les entomologistes disent qu'on diminue de façon très importante, mais on n'éradique pas un, il va toujours rester un fond de... Bon. Mais quand même, ça a l'air de marcher mieux d'ailleurs que Wolbachia, je crois. Par contre, pour ce qui concerne les culicoïdes, si on prend le cas de Ropouch, on n'a absolument aucun outil. Donc il faut continuer la lutte antivectorielle, en évitant d'utiliser évidemment des insecticides, mais en utilisant ces approches. Et il faut aussi, et ça rejoint la question de Claude, développer aussi des approches anti-vecteurs, mais pas uniquement moustiques, puisqu'on risque d'avoir... on n'a rien pour les tiques, par exemple, on n'a rien pour les moucherons, et on a quelques outils pour les moustiques, mais probablement encore à améliorer.
Merci beaucoup. Je voulais revenir sur la fièvre hémorragique de Crimée-Congo. Est-ce qu'elle peut être confondue avec la fièvre hémorragique avec syndrome rénal des hantavirus?
Oui, c'est une bonne question. Alors sur le plan clinique, j'en sais trop rien, je vous avoue. Bon, la fièvre hémorragique syndrome rénal, elle est plutôt présente dans le nord de l'Europe ou l'Est, etc. Là, on est plutôt dans le sud. Sur le plan clinique, je ne sais pas, mais sur le plan épidémiologique, c'est quand même assez lié à la présence de tiques. Donc, on a un effet saisonnier peut-être plus important que dans le cas de la fièvre syndrome rénal. Et c'est effectivement lié aujourd'hui aux régions dans lesquelles les personnes ont pu être infectées. Après, sur le plan clinique, je ne peux pas vous répondre.
Plus de questions dans la salle. Y a-t-il des questions sur Internet? Non. Alors, merci beaucoup pour votre présentation.
« 25 à 50% de la variabilité des maladies complexes humaines et d'origine héréditaire, impliquant des milliers de variants à effet cumulé. C'est ce que montrent les études d'associations pangénomiques. Grâce au progrès du séquençage et de l'imputation génétique, le risque individuel peut désormais être évalué. Cette approche du score de risque polygénique est aujourd'hui testée en médecine humaine au CHU de Liège. Monsieur le Président, c'est de cela que va nous entretenir le professeur Georges, membre bien connu de la RMB.
Oui, donc nous avons le plaisir d'accueillir le professeur Michel Georges, qui est vétérinaire de l'Université de Liège et qui a un master en biologie moléculaire de l'Université libre de Bruxelles, où il a travaillé dans le laboratoire du professeur Vassar. Il est professeur aussi de génétique humaine à Salt Lake City, dans l'Utah. Donc il est toujours, je pense, professeur de génétique et génomique à l'Université de Liège. Et il a joué un rôle important dans la fondation de l'Institut GIGA, dont il a été directeur de 2016 à 2023. Il a reçu de nombreux prix comme le prix Wolf Prize, le prix Francky en sciences biomédicales, et il est membre aussi de la National Academy of Sciences aux États-Unis. Et nous allons l'écouter avec attention.
Merci beaucoup. Donc le West Nile virus... C'est moi qui dois aller chercher les médias, je suppose. Où est ma présentation? Au secours. C'est même pas un Mac. Donc en fait, vous allez découvrir que dans un élan de lucidité, j'ai changé mon titre. C'était Polygenic Risk Score State of the Art, et maintenant c'est A Primer. Je me suis rendu compte cette semaine que c'était plus approprié. Ça c'est le mien. Bravo. Allons-y. Ah, c'était la suite. Ah, c'est pour ça que vous nous demandez d'envoyer les présentations à l'avance. Merci. Donc ce que je me propose de faire, c'est d'abord être sûr qu'on comprenne tous de quoi il s'agit. Sorry, I'm going to switch to English. So that we are all on the same wavelength when it comes sort of for the basis to understand what polygenic risk scores are. And I think to do that, I first need to say a few words about genetic polymorphism. You'll tell me that's maybe a little bit too low, but I think it's still worth it. So first of all, in the room here, if we sequence the genome of everybody, there are a bazillion genetic variants, of which the most numerous ones are called SNPs, and these are the ones that are represented on the right-hand side of the slide there. And if you have a SNP and you look at the whole room here, you can count how many of the chromosomes have, let's say, the G allele versus the T allele. So you have a frequency of the two alleles, and obviously one will be more frequent than the other one being the minor, and so you have the so-called minor allele frequency, which can go from 0 to 0.5 to 50%. And if we study the genome of all the people in this room, we're going to find, as I said, lots of these polymorphisms, And if I sort them based on this frequency, distinguishing things that are quite common in the population, their frequency being more than 5%, we will, in the out of Africa population, find around 10 million of these things. But if we now go and look at the ones that are rare, less than 5%, people say 100 million, but there is more than that in the population. So the first thing to know, genetic variants. SNPs are the most numerous. They are characterized by a thing called minor allele frequency. There are lots of them, and we can distinguish common ones, 10 million in our population approximately, versus rare ones. The minor allele frequency, from a biological point of view, has something interesting, is that there is a relationship between that frequency and the age of this polymorphism. So polymorphisms have, or variants, have a life, they are born, then they live in the population, and then potentially they die or get fixed, which is close to the same. And so if a polymorphism is frequent in a population, it means that a long time has happened between its birth, so it's an old thing, while if it's a rare one, it will most of the time be a relatively recent one. But that has big implication for what I will tell you at the end. Common variants are shared between different ethnic populations, and rare variants are more often population specific. If we take the number of polymorphisms and the distribution of the frequency together, the so-called nucleotide diversity in our population is approximately 1 in 1,000, meaning that if I take 2 chromosomes and I read them, I will find a difference approximately 1 every 1,000 base pairs. And if I compare that with the similar exercise done between a Homo sapiens chromosome and a chimpanzee chromosome, I'm closer to 1%. All these polymorphisms, approximately, I would say 99% of them, have no impact on who you are, on your phenotype. As Leif was saying, they're neutral. But so we estimate that of the order of 1% affect the function of at least one gene and may affect the phenotype. So they're so-called functional polymorphism, which they can do either by changing the sequence of a gene, typically a protein sequence, or by affecting its pattern of expression, regulatory variants. 2, notion of heritability. First of all, what is the heritability? You have to go a little bit in statistics and remind yourself of what the notion of variance is. So the heritability is the proportion of the variance for a trait that is due to differences in the genotypes between individuals. So imagine you take height. We don't all have the same height. We can quantitate the difference between individuals in the population as the variance. Part of that is due to differences in genes, and part of that is due to differences in environment. So we measure the true variance, and now we do an imaginary experiment. Imagine that you neutralize everything that is environment. So imagine that everybody grows up in exactly the same environment. Well, suddenly the variance will diminish. The ratio between the variance without environment and the observed variance is the heritability of the trait. So how is this thing measured? In the past, I would say it was done by twin studies, and I don't have time, but just by looking at these pictures, some are dizygotic and the others are monozygotic twins, you will see who the monozygotic twins are, and you will in fact have estimated heritability visually. That's the way it was done. Most recently, since we can sequence our genomes, there is another way of looking at the heritability which is unbiased and which exploits what is called Mendelian segregation. It's in fact the knowledge that amongst SIPs, we are all relatively alike amongst brothers and sisters, but some pairs of brothers or sisters are more alike genetically than other ones. And so we look at the correlation between the similarity at the genome level and the similarity at the phenotypic level across large cohorts, and it gives us a new way of estimating the heritability. But I think the important thing, at least for me, is the result of that. So we're right in the nature-nurture debate, and of course we all have our ideas about that, but the column there, which is called H2L, for heritability of the liability, gives you the estimate of the heritability based on twin studies for a whole bunch of pathologies that you might be interested in. And I think it's quite striking that values above 50% are nearly the rule, are very, very common. So I have the chance, if I may say, to study Crohn's disease, heritability 70%. As a veterinarian, these are things that we virtually never see. We work in the veterinary fields with heritabilities between 10% and 40, 50%. In humans, contrary to what I would have sort of spontaneously thought, you work with heritabilities that tend to be higher. So saying that a big part of the phenotypic variance is due to differences between genotypes of individuals doesn't tell us about point 3, what is the genetic architecture. So we talk about the genotypes, for instance, of how many genes. And so a bit caricatural, so we can sort of say we have a certain number of phenotypes that are monogenic, also called Mendelian traits. So there, essentially, to understand that, you just need to know what's happening at one gene to be able to predict exactly what the phenotype of the individuals will be. And I think that's something that is maybe not commonly, if I may say so, in all humility, commonly appreciated enough in the medical fields, but to me, Mendelian traits are the exception and they are not the rule. The rule is what Galton proposed in the in the 19th century, is that the architecture of the majority of the traits is polygenic. And as the name implies, that means that you don't have to look at one gene, you have to look at more than one gene. And then the question is, what's behind poly? Is it 10? Is it 100? Is it 1,000? Is it 10,000? Knowing that we only have 20,000 genes in our genome. And the remarkable thing is that it looks like we really have to talk about the thousands. And again, for the medical field, when you say that there are lots of genes, it doesn't mean that a given phenotype is a constellation of multiple monogenic traits. It's the fact that in every individual, if you want, and especially every individual that is affected, to be affected, you need to have a constellation of alleles at many, many loci that determine what is called your liability, your chance or not to develop the disease. So many genes, and for every gene, there is, or many variants, I should say, and for every variant, there is an allele that confers a risk, but which is very small. So for those of you that are familiar with odds ratios or relative risk, it's sort of 1.1 and even 1.0 something. So they're very, very small effects, but these effects are cumulative, they're additive, and so they can determine or they can cause big differences between individuals. You'll tell me, how do you know these things? You know, how can you pretend that? And one of the sources, recent sources of information, is the results of GWAS. I think you've seen in Leif's presentation some GWAS. So for those not familiar, the x-axis is chromosomes, every dot is a variant, and the y-axis is evidence that this variant plays a role in a certain disease. And you see a little bit the evolution over the last, let's say, 10, 15 years, when experiments were done with 1,000 individuals, tens to 100,000 of individuals, and more recently, for height, millions of individuals. And essentially, the conclusion for height today is 12,000 variants affecting the function of 1 out of 4 of our genes. So this polygenicity is still a matter of discussion. It's actually interesting if you make the connection with the herring, and we may have a chance to talk about that over coffee, but today a number of people will tell you it's not polygenic, it's omnigenic, which is a term that actually has been used by Jonathan Pritchard. Another thing which is quite interesting, I think, if you think about all these effects, is that there is a relationship between the minor allele frequency that I introduced and the size of the effect. Typically, things that are rare have a bigger effect than things that are common in the population. And you would say, you know, what the hell, where is that coming from? And at the present time, the best hypothesis is that this is a signature of selection. So big effects tend to be most of the time deleterious or mildly deleterious, and that pushes the frequency of these alleles down in the population. Okay, so now I think you have everything I think you need to know to sort of get into PRS. But being a veterinarian, I'm going to say breeding value first, because the notion of PRS is a notion that, again, exists since the beginning of the 20th century, and we've called that the breeding value. And so what is the breeding value? It is the sum of all the allelic effects at all the loci which are affecting the trait. So if you have a polygenic trait that would be affected by, say, 100 loci, 100 variants, you essentially count the number of variants that is, let's say, increasing the trait. And if you want to do it very well, you not only count the number, but you weight them by the size of the effect. And you're got, so you know all the loci, and you sum these things, and you get a value, and that's what animal breeders call the breeding value. And they have developed, during the 20th century, methods to compute these things. And a big breakthrough was the individual animal model. This is statistics. For those of you that like statistics, so we are in linear models, we're in mixed models, which means that some of the effects are modeled as random effect, and the individual animal effect is a random effect. And essentially when you do that, which is really a machine learning method that was developed before the term machine learning existed, estimates the breeding value of individuals with a remarkably high accuracy. Now, to do that, you need to understand two things. One is animal breeders believed very early on in this polygenic, not to say omnigenic model, and a very famous guy called R.A. Fisher brought it to the extreme, and he said the infinitesimal model. So essentially what he said is, let's assume that every base pair in the genome contributes as much to the phenotype. So he said, now let's forget about localized effect and let's look at it like butter on bread. We spread the thing out evenly across the entire genome. If there are really thousands of loci, this infinitesimal model is a very nice approximation, okay? And then you can forget about individual loci, and the only thing which is important is to know how much of your genome, how much of this infinitesimal genome you share with all the other people in the room. So this is what is called the relationship matrix. So imagine that we want to calculate the breeding value for the auditorium. We would need to compute the relationship between all the individuals in the room, and then within this mathematical framework, it allows us to estimate the breeding values very accurately, just from knowing the kinship relationship between all the pairs of individuals in the population. And this was done until a few years ago, just based on genealogy. That's why breeders are crazy about genealogies. You know, they know the mothers of all, the grandfathers of all their cows, and it's registered in detail, and it's allowed to do that. And it has really sort of had a major impact on the cost of the things you buy in Carrefour, to some extent. Now, so this is what happened from 1960, I'm not kidding actually, from 1960 to 2007. And in 2007, the kinship relationship, this matrix, this relationship matrix, was not anymore computed from genealogy, but was computed from looking just at reading the DNA. And which is obviously where you would have to look to sort of, where the relationship matrix is materialized. And so this has generated the so-called genomic selection revolution. So from the moment that people did that, they said they didn't say anymore we compute breeding values, they said we compute genomic breeding values, and it has had an incredible impact on response to selection. So just look at these blue curves here. This is 2007, and it's for a bunch of traits that people select from, and so you see every time this break in the curve, it's either going up very quickly, or if you select against something, it's going down very quickly. But it has had an incredible impact in agriculture, and it has been very quickly adopted after animals by plant breeding. To give you an example, just in our small lab alone in Liège, we have now genotyped 1 million, Wouter Koppieters and his team, 1 million cows. We have whole genome sequences of thousands of cows. It's easier for me to get money to sequence and genotype a cow than a human. I have no clue why, but it's the fact, right? In the Walloon region, many more cows are genotyped than humans. In the Netherlands, every calf that will be born, people will look at its DNA. So it's truly a remarkable, once-in-a-lifetime revolution that we have witnessed with a major impact on genomic progress. Okay, polygenic risk scores. It's the same, right? It's the sum, so it's the sum of allelic effects, obviously not on milk production, but on the effect on the chance or the risk to develop a certain disease. But in terms of principle, it is exactly the same thing. So what I put in the slide here is selected. So to the best of my knowledge, very few, if anyone, has tried the old-fashioned, you could say, method based on the gametic relationship matrix in humans. And we could sort of think about why that is. So people have sort of gone for the godlike approach, which is imagine that I knew all the loci and I have all the alleles. I just have to make the sum of the allelic effects, okay? The problem is that we don't know all the loci. So essentially what is happening now when people compute PRS is they go to these GWAS plots and they start at the top and they progressively go down. So they take the most strong signals and they say these ones, everybody agrees that we use them to compute the PRS. Everybody says yes, of course. I mean, these are very convincing. This one we are going to take. But, you know, it's a polygenic trait and I'm only using 4 top. Okay, so let's go a little bit deeper. And so then you go deeper and deeper and deeper and deeper, on the one hand, to calculate your polygenic risk score, but another risk, not the disease risk, but the statistical risk of bringing in false positives increase as you go down. So it's sort of a fight against noise as you go down to try to get a nice picture of the polygenic risk score. So you can do 2 things, you know, you can put more individuals in your experiment, so the peaks are going up, or you can ask the engineers to come up with machine learning methods to try to go deeper and try to minimize the noise that you bring. So that's what's happening. And essentially what you do, if you compute these polygenic risk scores, these breeding values, is you stratify your population for the risk to develop the disease. It's called, classically, the liability. And this thing has a normal distribution, le théorème centrale limite. If something is the sum of many things, it's automatically giving you a normal distribution. And so you start to have the capacity to differentiate people that are genetically at high risk versus those that are at less or very low risk. I think to be more practical, this is, I think, an informative slide. It shows you the distribution in a human population for breast cancer of women between the age of 30 and 80. When you don't use genetics, you just use the standard, let's say, risk factors for breast cancer. You get this gray curve, right? If you now add or you compute the polygenic risk score for breast cancer, what is happening is that the curve is spread out. So you start to differentiate people much better than you did without. You can identify individuals at very high risk and identify individuals at very low risk that were noyés dans la masse before. Okay. More and more people are obviously going into this thing, and so this is an example where you can now go on the web and you can bring in SNPs and they compute your polygenic risk score given the model you choose for whatever you want. Very good. This being said, and Liège, I'll tell you at the last slide what we do in Liège, but when compared to what I have witnessed in animals, nothing is happening in human. You know, it's like snail pace. You know, so PRS are absolutely not adopted eagerly by the medical profession until now. So why is that? So I was sort of thinking this week, and that's why when I changed state of the art to primer, but why the hell would that be? Why is it that something that from a biological point of view seemed to be very similar has had a tremendous impact in one field and doesn't have so far a big impact in a neighboring field? And I thought, well, actually maybe that's not so much science, So proposition one, the simple economic driver that existed in animal breeding does not exist in human medicine. I think everything was ready in animal breeding, not only technologically, but also in terms of economics, to get this thing going. There were, in this area, there were people that could really make profit out of this thing, or if they were to decide not to use it, they would die. They have died. So there was a very strong incentive. And I asked I allow myself to put in between brackets something for the Académie de Médecine. Which actor benefits from an increase in efficiency of the medical system? Just a thought for over beer, okay? Proposition two, well-organized, curated, easily accessible, unbiased health record databases do not exist in most countries. So if you look at animal breeding, it's extraordinary how in many countries things are well organized. Someone is going to say hello to every cow once a month, if not more, and recording plenty of things and putting that into databases, which are organized in exactly the same way all over the world. And so these databases were just ready to be connected immediately and effectively to genotypes. And I don't know what your experience is, but my experience with electronic medical records, including in Belgium, it's a catastrophe. C'est le bordel, okay? So I think that's also one of the reasons why it's not so easy to implement these methods, because you need to have access to well-organized electronic medical records, and they are not there, okay? So which are the countries that are the most advanced with the polygenic risk scores? It's the UK, with the UK Biobank. It's the countries around the Baltic, so Scandinavian countries and the Baltic countries. And if you go look at the latest issue of Nature, Taiwan and Japan have so Taiwan is now having something that is close to equivalent to the UK Biobank. Okay, but then there are certainly also not economic or let's say factors, so maybe closer to science. So PRS are not diagnostic. So in animals, we're not interested very much in the well-being of one animal. We are interested in the productivity of a herd. And so we make predictions about the average of what is happening for 100, 200, or 1,000 animals. In humans, it depends a little bit where you come from, but depending on your culture, it's most of the time very individual-centric, and which is good, probably, we can discuss that. But I think that's also one of the reasons why there is a barrier. And I try to illustrate that by two examples that I simulated a long time ago. One for a continuous trait and one for a discrete trait like a disease. So I imagined I stimulate in the computer height, and I imagine that height has a heritability of 85%, which is close to the reality. And so I simulate a population that has, let's say, a medium height, I think I did, of 1.70 meters. And when I add the noise due to the genes and the environment, the average distance from this moyenne was, let's say, 11 centimeters, okay? So essentially what you could sort of say, I take an individual at random out of the auditorium, I don't receive any of its DNA, I can't do anything, and they ask me, it's my exam, what is the height of this person? Well, the best you can do is give the mean, right? So you say 1.70 meters, and the mistake you make on average is 11 centimeters, okay? So now imagine that you have a PRS for height. And not only, so they say this individual, they don't tell you the height, but they now say, they give you DNA, and you look at the DNA and you predict the genetic height. And so I sort of looked at either I'm really got and I know all the genes, or, which is the case now for most of the train, let's imagine that I know half of them. I explain half the heritability. What is happening? Well, when you explain half the heritability, your standard error is going from 11 to 8. And if you know everything, your standard error is going from 11 to 4, okay? So just to tell you that even if the heritability is high, there's still a lot of environment in a way. And so even if you are got, you will still make a mistake in estimating the height. And on your way to be a got, where we are now, half, you know, the advantage of the polygenic risk score is not very high. So probably quantitative traits are not so important for the doctors here. So if I look at a binary trait, let's say diseased or healthy, I took 60% heritability because it's closer to the reality, and I explain half the heritability or the whole heritability. And then I imagine, you know, why the hell would we do a thing like that? Well, I suppose I'm going to take a certain, the most at-risk individuals, and I'm going to propose an intervention or a screening or something like that. So what this graph is trying to show you, so imagine that you take the top 10 of the population in terms of risk, and you say, you guys are going to take statins, or you're going to go more often for a mammography screening, or whatever you're going to tell them. Ultimately, the question is, of all the people that in the end would do the disease, to how many do I propose this, what I hope will be good for them, right? So if you take the top 10, how many of the individuals that are truly at risk, that are going to develop the disease, are taken care of by my intervention, okay? And then the other thing is, of all the people that I'm saying, now you're going to take statins, how many actually needed it? And so the red curve is showing you for two scenarios what proportion of the people that need to be caught are caught, and what proportion of the people that are caught actually needed it. And I'll give you an example for 1 in 250 Crohn's disease. At 10%, I capture pretty much everybody that is going to develop disease. If I know everything, if I'm got, and sort of today it would be 70%. But the problem is that out of all the people to which I would impose the treatment or suggest the treatment, only 5% actually needed it. The vast majority didn't need it because it's a rare disease, okay? And so there you have another scenario for type 2 diabetes. And so what you see is that it's not, PRS are not diagnostic. You make substantial errors in both directions. You don't capture everybody that should be captured, and of the people that you capture, many actually don't need to be there. Now, this being said, so it's a valid criticism of PRS, but doctors use these things all the time, right? So while I was preparing here, I was reading the way people sort of prescribe statins to people based on their risk profile to do cardiovascular diseases. And I sort of hear that an estimate of 20%, I think, risk from the moment you do the diagnostic justifies the prescription of statins. But if you look at that, you sort of realize that by doing that, you, quote unquote, save 2 people out of the 100 to which you prescribed statin. So you actually have an impact on 2 out of 100 people, and 1 out of 100 will develop type 1 diabetes because of the statins. So PRS have a problem, but it's statistics, if you will, that is already part of the clinical activity today. Another problem which is important for humans is that genetics has been very much Eurocentric so far. So all the GWAS have been done on European population. And as a consequence, when you compute PRS today, you compute it based on European data, which are very poor predictors of other ethnic groups. So the implementation of PRS creates a an inequity, an important inequity problem. Obviously, PRS raise GDR concerns. I don't know whether I still have time. I have 3 examples just to say why I think you will ultimately use it anyhow. So the first is, there exists monogenic forms of common complex diseases that confirm relative risk of 3 or more, 4 or 5. And examples which you know of are BRCA1 and BRCA2 mutations for breast cancer, LDLR and at least 2 other genes for coronary artery disease. And today, the diagnosis for these mutations is used in your clinics. It's used by my colleague Vincent Bourse. So if you have a family in which you have BRCA mutations, these people are going to be given a special protocol of treatment. Well, today, even if you eliminate from your analysis sample the people that are at high risk because of these monogenic forms, if you just do PRS, you will identify individuals that are at the same risk than the one confirmed by these monogenic forms in higher number, okay? So go read the paper by Kera et al. 2018. It sort of shows that if you were to do PRS, you would capture, so you would identify for approximately 1 in 5 people out of the room a relative risk of 3 or more for one of these diseases there. So we use genetics in the monogenic Mendelian context to take care of a fraction of the population in a specific way that everybody seems to accept. We can directly do the same thing using PRS because we will identify individuals that have exactly the same risk profile, but which has a polygenic architecture and not this monogenic architecture. And because PRS are generic, you do the same analysis for every disease, you can actually immediately compute it for multiple diseases. So example 2, and so I thought about, I think that there's something important in there and that I think will explain or justify the fact that it will be used. The efficiency of preventive treatments. So when you look at the literature, you have, in the risk factors, you have non-modifiable risk factors, that's for instance the genes, and you have modifiable risk factors. That includes behavior, screening, treatment, and so on. And so what you see is that in the studies that are being published, you see that the efficacy of pretty much all the modifiable treatments, the preventive treatments, are more efficient in the people at high risk than in the people at low risk. So this is an example where in 3 studies, people look a posteriori at the beneficial effect of statin use in individuals sorted by their PRS score. And the fact that the red bar is considerably higher on the right-hand side means that the beneficial impact of statin is much higher in this high genetic group. And the slide on the right is telling you the same story for breast cancer. But so essentially, whether you are going to use a preventive action, whatever it is, on a patient or not, depends on the cost-benefit analysis, where cost is economic, but also medical, the risk you take. And so what essentially these results are telling us, that this ratio is really in favor of using the PRS because it identifies the individuals in which it makes most sense to use the preventive factors. So PRS says risk, polygenic risk score. But if I go and talk to my colleague and friend Edouard Louis, who is a gastroenterologist, he tells me, listen, I don't need a PRS to tell me whether this patient is Crohn's disease or ulcerative colitis. I need a PRS to tell me whether the patient will have a smoldering, if I may say, so I think that will remain chronic, or whether he will flare, or whether he's going in a direction that requires surgical intervention. Or he, of course, people would like to know what's the chance that this patient will respond to this medication versus another? Will be there deleterious impacts of the drug? Well, all these questions equate to traits which have a heritability, and if you have enough data, they are amenable to the same kind of analysis as what is done with the polygenic risk score. So the scope of PRS exceeds by far just the risk to develop a disease. They can be combined with classical risk scores, things that are done in Finland. In light of all these things, and essentially to a large extent our experience in animals, at least to some extent, we sort of thought at the University of Liège that, especially when we were looking at users of FEDER funding, that actually use that to engage into a project that would position us in a way that we could try to deploy or at least test PRS in the clinic would make sense, okay? There was sort of a scientific progress that we thought was promising, and we thought it was a very ethical use of money that is devoted to try to develop structure that will enable many people to benefit from it. We thought that it was a structuring project for the medical community, and so we have oriented at least part of the funding into setting up a project like that, and we've called this project Ensemble. Together, and I would say there are many people that have been involved into that. Vincent Doriot is here, Edouard Louis has played a key role, Vincent Bourse, and many, many others. And so at the present time, we are approaching, so then you say, how do I start a thing like that? And as I said before, what you need is big databases of where you not only have DNA, but you also have the electronic medical records organized and attached to it. And at the present time, we are approaching the 10,000 individuals in Liège thanks to that support. So this is dwarfed by the million in the UK, the 500,000 in Finland, the 500,000 in Taiwan. But at least they allow you to test every PRS in your population. So what people call calibrating the PRS. So the main motivation for that is sort of to say, for our clinicians to be in a position to sort of test it, they at least need to be able to see whether the PRS computation generates the expected shift in the distribution between the controls and their cases. And so these things are going on. A number of years ago, I said that. I'm not sure I would dare to say it again, but I think there is an element of that. It's the fact that I think that it's very likely that the PRS are going to be given to clinicians. And the question is, do you buy them from Google, or do you want to be an actor into sort of developing them? Also, if you want to use them well, you have to be prepared and you have to have played with these things in advance. So thank you very much.
Une première question, là.
Est-ce que je peux demander à chacun, s'il vous plaît, en accord évidemment et avec l'autorisation du secrétaire perpétuel, de donner son nom en énonçant sa question. Ça permet aux gens qui sont sur Internet de savoir qui parle. Merci.
Bonjour, Bernard ANSES, je travaille sur la maladie d'Alzheimer. Merci pour cet exposé brillant des PRS, et je vous rejoins dans votre hypothèse numéro 3 qu'en médecine humaine, il faut un intérêt individuel pour qu'à un moment donné, les collectivités se mobilisent. Et dans mon domaine en particulier, mais à mon avis dans beaucoup d'autres, on avance vers des PRS qui, dans certains cas, se rapprochent des prédictions monogéniques. Dans le cas de la maladie d'Alzheimer, on a identifié des PRS qui donnent 100% de risque de développer la maladie. L'unique différence avec les aspects monogéniques, c'est qu'il s'agit d'une combinaison de plusieurs gènes et pas d'un seul. Nos confrères généticiens humains, ce qui n'est pas votre cas, refusent en fait de faire de la prédiction chez leurs enfants. Pourquoi? Parce que ça reviendrait à dévoiler la présence d'un seul de ces gènes. La probabilité que la même combinaison soit passée à l'enfant est en fait peu vraisemblable. Si on parle d'une dizaine de gènes, on peut calculer la probabilité, mais il est plus probable que chacun des enfants en ait pris un certain nombre et pas l'exacte même combinaison, et donc le risque ne sera plus de 100%, mais d'un autre. En médecine, on aime bien le diagnostic, le noir, le blanc, on est malade, on n'est pas malade. Doit-on évoluer, nous, l'ensemble de la communauté médicale, vers une gestion différenciée, les 1000 nuances de gris, et donc arriver quelque part à un bénéfice individuel? Peut-on envisager un bénéfice individuel de cette polygénétique qui va devoir amener une gestion de risque et pas une certitude?
Ok, mais donc vous savez mieux répondre à toutes vos questions que je ne le pourrais, mais je vais quand même essayer de... Donc ce que vos questions suggèrent, du moins, ou me font dire, c'est cette notion, je ne l'ai pas mentionnée, j'aurais dû, de actionable. Donc, c'est vrai que ça n'a pas énormément de sens, et ça je peux comprendre, d'aller faire des prédictions pour des risques de pathologie, où la seule chose que l'on peut offrir au patient, c'est une évaluation de son risque et aucune intervention qui va l'aider. Mais je crois que ça, c'est quelque chose qui est, en général, j'ai presque envie de dire respecté, c'est-à-dire qu'on limite ce genre d'informations quand on a la possibilité de proposer quelque chose qui va améliorer la vie du patient. Alors, donc en parenthèse, là, quand vous dites on a des PRS qui sont associés à une probabilité de 100% de faire la maladie d'Alzheimer, donc ça, ce serait intéressant qu'on discute en détail, parce que c'est quelque chose que des ingénieurs, aujourd'hui, quand ils rentrent en génétique, nous disent souvent. Leurs méthodes sont extraordinaires, elles classent parfaitement, et on leur dit l'héritabilité est de 50%. Tu ne sais pas tout prédire. Mais maintenant, ça dépend évidemment comment on... Donc, il faudrait rentrer dans les détails. Mais c'est un peu pour ça que j'ai essayé de montrer les dias, c'est que la génétique ne peut prédire que la partie génétique, et aussi longtemps qu'on ne connaît pas tout, la projection est fortement affectée. Donc en fait, ce n'est pas linéaire, ce n'est pas comme si on explique la moitié de la variance, on est à mi-chemin par rapport à la prédiction correcte. En fait, c'est quelque chose qui monte de façon exponentielle. Sinon, est-ce que, donc si j'ai bien compris la troisième partie de la question, donc est-ce que les médecins doivent devenir gris plutôt que rester dans le noir et le blanc? Mais en fait, je crois qu'ils le sont déjà. C'est pour ça que j'ai... Donc je crois que le médecin utilise déjà dans son arsenal, je suis très mal placé pour le savoir, mais vous me direz, donc je crois qu'il utilise déjà des protocoles dans lesquels en fait on fait des calculs de risque que l'on combine de façon bayésienne ou non pour arriver à la meilleure décision possible en fonction d'un certain nombre de critères qui sont, comme vous l'avez dit, en général pour l'instant des critères individuels. Alors, est-ce que la médecine doit uniquement être individuelle? Bon, ça, moi, j'aime quand même bien ma proposition 1. Il y a un problème avec le système médical à l'heure actuelle. Il n'y a personne qui a un intérêt à réduire les frais du système médical. Non? Ça ne peut pas durer, ce truc. Voilà.
I have a question. So I like very much your picture, one of your first slides, where you compare monozygotic twins and dizygotic twins. And what you immediately see there is that the dizygotic twins vary much more than the monozygotic twins. But that is a conflict with the infinitesimal model, right?
No.
And what you've been saying about the thousands of genes affecting stature, for instance. So how do you explain that?
Yeah, so that's a very common thing. So for instance, the doctors tell me I already put familial risk in my model, which is very often the case. And then if you put familial risk, what is the added value of putting polygenic risk scores, and I think it's related to that. And so if you go in classic textbooks, Faulkner, the genetic variation that still exists between full SIPs is half of the genetic variance. So quite surprisingly, within families, you still have half of the genetic variation that exists within the whole population. I could do the demonstration mathematically, but so there is still a lot of genetic variation within families. And that's why it's so efficient, that's why genomic selection is so efficient.
Yeah. But if there were thousands of genes with all with a tiny effect, they should be quite even.
Yeah, I know. Okay, so typically the demonstration of that sort of the mathematical demonstration is far from the biology. So I think probably you would like, for instance, us to go and look at the number of chromosomes maybe. So what you say is the number of chromosomes was infinitely large, then the segregation around 50% should be virtually zero. If they are all on one chromosome, suddenly you have more fluctuation. But usually people have not included that in sort of I'm not aware of anyone saying the residual genetic variation within full SIP families is a function of the number of chromosomes, but we could look at it.
Just to comment on that, and Jonathan Doufis from the University of Namur, in a certain context, especially when we were discussing about family history in my field, for example, only 15% of the family, 50% of the family history can explain, for example, the generation of a thrombotic event, while thrombophilia screening, so it's not really polygenic risk factors, but if you are adding different mutations, can predict up to 60% of these risks. So I really think that if we're able to provide polygenic risk factor in addition to family risk factor, obviously you will increase your probability of detecting something, and at the end you can act by, for example, providing other treatment or having other habits with the patient. So I really think that here, just to comment also on what Bernard mentioned, it's quite important to have both the clinics and also polygenic risk factor when we are treating patients.
I know I should have emphasized that. So nobody that is computing polygenic risk scores want to replace the non-genetic risk factors. You always bring them in. And so mathematically there is no problem at all so you build on top. And in general, people show that the added value of the PRS is quite substantial.
Here, it's not really substantial because we can increase by 55%. We have 55% more detection of people at risk. So it's really, really important. And
but that depends on where you put your threshold, right?
Yeah.
So here you say I increase by 50%, but if I tell you, okay, how did you define the ones at risk? You take in a threshold. And your 55%, je peux lui faire dire ce que je veux en jouant with the threshold. You see?
Yeah. Here but the thing is that here we have identified very specific mutations that are linked to that. But another problem I can see from my practice is that the difficulty is to implement that in clinical practice. And especially because people are speaking also about the cost of screening. And I think when we are discussing about that, it is quite interesting to see how much event we can avoid by using this technology. So really have a long-term cost-effectiveness view about this screening instead of just viewing maybe in 1 year of very direct cost, because when we have a disease, it's not only direct cost of treating a disease. Sometimes it's also indirect cost, like the loss of productivity and so on. And I think it's important to implement that also in the cost-effectiveness models.
I agree. So yeah, I agree. So, and then you actually, to some extent, go a little bit away from the individual interest focused only. When it comes to cost, for us veterinarians, it's really funny, right? So do you know how much? So we genotype this 1 million cows. Do you know how much it costs to genotype a cow? The chip is €5 and or less, two something euro, and so the total cost of genotyping a cow is less than. That's the price that the farmer pays. It's less than €20. And if you ask a doctor to do the same thing for a human, he will charge 500 €. And then you tell me, you know,
at least.
Oh, now I understand how he got this big villa. Yeah.
On a une question du professeur Knopp également.
Miriam Knopp, beautiful talk. Thank you so much. I want to ask you something about actionable PRSs and other genetic diagnoses. So obviously, you know, it's difficult when you have your whole genome sequence to hear that you're gonna develop young onset Alzheimer's disease or something. Because there is not much that can be done about this. But I think, and so for that reason, obviously, you know, people consent, they want to know if they're going to be informed even of actionable things, yes or no, because they have the right to refuse that. But I was thinking back of a friend that I referred to, a neurologist, number of years ago, who was diagnosed then with ALS, with amyotrophic lateral sclerosis, and well, the neurologist who made the diagnosis communicated this to the patient, and I said, well, there is not much to be done to the neurologist now with this diagnosis, and he said, that's not true. You know, there is a lot that needs to be put in place, because obviously there's nothing we can do about the course of the disease, but then there is a lot that needs to be, you know, put in place for this patient, you know, to live the few years that are still ahead. And I think perhaps we need to change the way we think about what is actionable and non-actionable, because even if it's non-actionable, there is still a lot that needs to be done by the person who has such a high-risk PRS for a disease. I just wanted, since you're a veterinarian, I thought you would be the good person to ask this question.
But I'm an even worse veterinarian than I am. I have a paper somewhere in the drawer saying that I'm a veterinarian, but that's all that's left, you know. So no, but I think so. This sort of brings two things to me. So one is I from now having sort of interacted with the different departments at the hospital, you sort of see that the potential use or not of PRS is really completely field specific. So, and I think every specialty will have sort of to find its way of making these PRS beneficial or concluding that for them they are not. That's so on the doctor side. And on the, let's say, population and because I suppose to some extent the comment you make, the comment could have come from a non-doctor. I mean, it's sort of a, and I think our attitude towards PRS and sort of getting used to live with what our DNA may tell us, including about our future, is something that we will have to learn with, I think, and it will change over time. So, and it's probably, but I could be completely wrong, but it's probably a bit paranoid now.
Il n'y a plus de questions dans la salle. Je suppose qu'il n'y en a pas non plus sur Internet. Nous allons passer à la dernière présentation.
Prescribing practices for the elderly in nursing homes have raised concerns regarding the quality of care. While a strong regulatory framework appears to be effective, several trials have shown that collaboration between clinicians and pharmacists can further improve medication safety and reduce inappropriate prescribing. In the future, developing core outcome sets could help systematically measure other outcomes that truly matter to both patients and caregivers. Mr. President, this is what Professor Hughes, foreign member of the Academy, will now focus on.
Merci. Malheureusement, le professeur Carmel Hughes n'a pas pu se joindre à nous aujourd'hui pour des raisons familiales, mais nous avons enregistré sa présentation qui vous sera projetée dans un moment. Donc, le professeur Carmel Hughes est professeur de Primary Care Pharmacie à l'école de pharmacie du Queen's University, Belfast, en Irlande du Nord. Ses centres d'intérêt sont la prescription de médicaments à des personnes âgées. Elle a publié plus de 350 papiers sur le sujet. Et donc, s'il y a moyen d'avoir la présentation. Merci.
Thank you very much for this great honour, and I'm very sorry that I can't be with you today. My name is Carmel Hughes. I'm based at the School of Pharmacy at Queen's in Belfast, and the title of my presentation is Optimising Prescribing of Medicines in Older People, Approaches and Outcomes. So what I want to cover in this presentation are the challenges to prescribing in older people, the approaches that can be taken to try and improve the prescribing of medicines in older people, and what outcomes we should be seeking to measure in studies that are carried out to try and assess whether or not what we are doing is making any difference to the prescribing of medicines in older people. This quotation has been used extensively throughout the literature and really defines the issue around the use of medications, in that it is arguably the single most important healthcare intervention in the industrialised world, according to Jerry Avorn, a geriatrician who's based in the United States. And it really encapsulates the most important intervention that we need to think about when we are caring for older people. And it highlights as well that there are several challenges in prescribing for older people, the challenges being altered pharmacokinetics and pharmacodynamics. Many older people will have multiple medical conditions, which is described as multimorbidity, and because they have multiple medical conditions, this will also lead to polypharmacy, or the use of multiple medicines. And because they are taking many medicines, that in turn can also affect adherence. And finally, there are also important considerations as to the stopping and starting of medicines as well. One group of older people who have challenges with medicines are those who live in nursing homes, sometimes called care homes. The photographs on the left-hand side of the slide are those from the early 1990s in London, which show the precursor of nursing homes, where they were known as workhouses. Care homes have long been associated with poor quality of care, and this is highlighted by some of the newspaper headlines on the right-hand side of the slide, showing some of the media reports which have highlighted examples of such poor care, and indeed the deaths of some older people in care homes. Older people who live in care homes are characterised by complexity. And care homes as organisations in themselves are very complex organisations. They have high levels of interdependence and connectivity. They don't sit in isolation from the rest of the healthcare system. There are links with general practice, social work, hospitals, so that accounts for this interdependence and connectivity. There are many demands and changing demands within care homes. They can be unpredictable environments because older people are frail and can deteriorate quite quickly. And they are also personified by uncertainty as well because of the unpredictability. But they're also supposed to be acting as a home environment. This is where many older people will be living, and it is supposed to be their homes. Residents themselves are quite complex in that they will be frail, they will require several healthcare services, and therefore that leads to resource consumption, and they have needs for social support as well, such as eating, drinking, dressing themselves, and other activities of living difficulties. This slide highlights the complexity that older people again will demonstrate. This slide comes from a study that we carried out in a number of nursing homes in Northern Ireland, and you can see that the vast majority of older people were older than 80. They were predominantly female. They were living in a care home for around about 2.5 years. The diagnoses that were associated with care home residents were the types of conditions that you might expect to see in older people, such as stroke, osteoarthritis and dementia. The mean number of diagnoses was just over 4, and again lots of prescribing going on, with almost 11 medicines on average being prescribed for each older person. So therefore, how do you attempt to optimize or improve medicines use when you're dealing with such levels of complexity? And I have looked at two different approaches, legislation and regulation, and also collaboration as means to improve prescribing and the use of medicines in this particular environment. This was one of the early papers that I published looking at a particular approach that was used in the United States, whereby they had a very extensive legislative framework, and we published this paper to demonstrate to UK colleagues that, number one, that this legislative framework existed, and whether or not there were any lessons to be learned in terms of how to deal with the legacy of concerns around the quality of care for older people living in nursing homes. The legislation that I'm referring to is the Omnibus Budgetary Reconciliation Act of 1987, also known as OBRA 87, sometimes referred to as the Nursing Home Reform Act. And this sought to improve the quality of care in US nursing homes through regulations, inspections and enforcement. And one of the regulations was the clinical justification for the use of psychoactive medications, notably hypnotics, anxiolytics and antipsychotics. And these medicines had been used essentially to compensate for the lack of staff in care homes, and they were described as chemical restraints and were being used to sedate and subdue older people. And this legislation sought to improve the use of these medicines, and following the implementation of the regulations and legislations, it was found that yes, there was a reduction in the use of psychoactive medications, but was a reduction in the number of medications enough to bring about an improvement in the quality of care? Or can it actually bring about a clinical difference as well? And this comment was made in an editorial again by Jerry Gervitz and another Jerry Avorn, both geriatricians, who stated that there needed to be an examination of the clinical consequences of changes in prescribing rather than viewing the change to be an end in itself. In other words, was it enough simply to see changes in prescribing, or did it bring about any other differences as well? So using an international epidemiological database from 6 countries, we were able to show that the legislation present in the USA did appear to influence the prescribing of psychoactive medications compared to other countries that didn't have equivalent legislation. But although the prescribing of psychoactive medicines was less in the USA, residents in US nursing homes fell more when we would have expected them perhaps to fall less, as these medicines have been associated with falls. So that led us to think, is regulation enough to improve care? And the evidence suggests that a regulatory approach appears to work best in preventing poor quality care rather than being effective in promoting good quality care. And this paper on the left-hand side was a review article that we published that looked at a number of these issues, and it led us to think whether or not we needed to go beyond regulation, and should we be promoting collaboration between healthcare professionals in improving the use of medicines rather than only relying on regulation on its own. I have been involved in a trial that involved the implementation of a new model of care called the Fleetwood Project, or the Fleetwood Model of Care. And this model required pharmacists to visit care homes on a regular basis, to use an algorithm or a flowchart to assess appropriateness of the prescribing of antipsychotic, hypnotic and anxiolytic medications. The pharmacists needed to liaise with GPs and other healthcare professionals and to document everything that they did on a pharmaceutical care plan. And we used this model in a trial whereby those homes randomized to receive this model of care were assessed compared to homes that did not receive this model of care. And what we found was that after 1 year, the odds ratio of a resident receiving inappropriate psychoactive medication in an intervention home was dramatically reduced compared to residents in a control home. And this is shown on the right-hand side in the forest plot, where there was quite a marked effect in terms of prescribing. And what we also showed as well, that this was a cost-effective intervention. And we published our findings in 2 papers in the Journal of the American Geriatric Society. However, this model of care, in essence, was reactive in that pharmacists were visiting the homes after prescribing had taken place. So we began to think about, again, alternative ways in which prescribing could be improved, and this led to a second study which moved from the reactive context to the proactive context, in which pharmacists who were qualified to prescribe medicines were responsible for all prescribing in care homes. And this is a model of care that has been developed in the UK, where suitably qualified pharmacists can now indeed assume prescribing rights and be independent prescribers for a range of different conditions. And this led to the Care Homes Independent Pharmacist Prescribing Study, otherwise known as CHIPS. And this was a 5-year research programme aiming to run an RCT in care homes, whereby we compared pharmacist prescribing to usual care in care homes, and we carried this out across a number of different regions across the UK, including Norfolk, Leeds, Aberdeenshire and also Northern Ireland. We published our findings in the British Medical Journal, and what we found was that there was a reduction in drug burden index, which is really a measure of the number and the complexity of drugs that are being received. However, we didn't see a reduction in falls, and we didn't see any kind of effect in health-related quality of life, but the intervention was well received and it was also safe. That also led us to think about what kind of outcomes should we be focusing on in studies? What should we be measuring when we're conducting trials or other types of research in older people? I've been involved in an organisation called COMET, which stands for Core Outcome Measures in Effectiveness Trials. And this organisation seeks to develop what are known as core outcome sets, or COSs, which are a minimum set of outcomes to be measured in all trials conducted in a particular area of healthcare. So the focus is very much on what we should be measuring in similar trials, but increasingly there is also interest as well in how we are measuring those particular outcomes. We have developed a core outcome set to be used in effectiveness trials that are focusing on optimizing prescribing in older adults in care homes. And you can see again on the right hand side of the slide the types of outcomes that we are recommending that should be measured. Yes, there's a focus on prescribing, there's a focus on adverse drug events and also errors, but we're also interested in quality of life, falls, all-cause mortality and admissions to hospitals and associated costs. And I think this kind of work and thinking about outcomes irrespective of where we work in research, I think should be a major focus. Back in 2009, a former editor of the BMJ wrote, "Could the current economic crisis be the making of evidence-based medicine? Perhaps it's only now in this harshest of financial climates that we'll really have to put the evidence to work." And I think we need to consider this comment. I think it's as pertinent now as it was back in 2009, as we live in a time of great uncertainty, and that we really need to think about the types of studies that we do, the kinds of outcomes that we measure, in order to generate the evidence that is going to make a difference to the health of society. Thank you very much indeed for listening.
Et pour ceux qui y sont habitués, évidemment, c'est tout à fait normal. Pour ceux qui ne le sont pas, et comme il y a des nouveaux avec nous aujourd'hui, sachez que l'Académie introduit systématiquement des demandes d'accréditation auprès de l'INAMI. Et donc, si vous êtes membre de l'Académie ou médecin, que vous nous suivez ici ou sur les réseaux sociaux, eh bien, vous pouvez, ou sur Zoom, évidemment, vous pouvez nous envoyer vos demandes à l'adresse contact@armb.be et les informations à fournir, bien entendu, nom, prénom, numéro INAMI. Et nous allons maintenant, avec cela, mettre fin à la retransmission YouTube et poursuivre en comité secret. Avant cela, 2 petites précisions. Prochain rendez-vous le jeudi 23 octobre dès 9h30 pour un symposium consacré à l'enseignement de la médecine. Soyez nombreux, n'hésitez pas à venir. On aura le temps de se voir au cours des pauses. Il y en a une un peu plus longue le midi et donc c'est l'occasion de se retrouver et de discuter au-delà, d'apprendre beaucoup de choses car le symposium sera très intéressant. Samedi 22 novembre 2025, 10h, rendez-vous pour la séance publique ordinaire. Et nous allons maintenant vous dire au revoir. Monsieur le secrétaire perpétuel, vous allez nous faire part des communications de l'Académie et bien entendu, mettons fin au suspense puisque